In this video, we're going to learn about how our cells in our bodies can control the expression of their genes. So the key idea is that genes encode for proteins, and those proteins do the jobs of our cells, so they dictate cell function. Every body cell has a full set of DNA that can produce all the proteins needed to make your body. So your genome is your full blueprint for making you. It's all the directions for all the proteins your body needs. But each cell has a different type and job. So as a result, each cell can only express some of its genes. We call this idea differentiation. Cells have different jobs and therefore different structures. Differentiation. So how does differentiation occur? Well, a gene gets expressed when a protein is made using that gene's code. So here we can see that our liver cells and our neuron cells are expressing different genes. So let's take a look first at liver. One of the liver's jobs is to detoxify and break down alcohol. So your liver cells need the alcohol dehydrogenase gene turned on, so it can make this enzyme and your body can break down any alcohol you consume. But your liver cell doesn't make neurotransmitters. That's the job of a neuron. So a neuron will turn on the neurotransmitter gene to make neurotransmitters for use in communication between nerve cells or between nerves and muscle cells. But your neurons don't break down alcohol, so they have that one shut off. So you can see different cells are going to express different genes based on their structure and their function. So a cell's gene expression pattern is determined by lots of information, both inside the cell and outside the cell. So let's take a look at this picture as an example. Okay, what we see is this yellow molecule right here. That is a growth factor. And a growth factor is a signal protein, and it gets released by neighboring cells, and it can trigger mitosis within the cell that receives it. So this growth factor is an external signal. It's something that is telling the cell, hey, it's time for you to turn certain genes on. So in this case, with a growth factor, you can go ahead and divide by mitosis. Now within the cell, there are additional proteins that have to relay this message. So we see there's like this chain of events. It's something called a signal transduction pathway. And in the end, we produce internal signals called transcription factors. And transcription factors are what are actually going to control transcription. They can say, hey, it's time to turn on the division promoting gene or turn on the growth promoting gene, those proto-oncogenes, so that the cell can then progress through the cell cycle. So your cells are constantly monitoring their external environment as well as what's going on internally. The right amount of different proteins or different sugars, how much energy is being produced, all these things are being monitored so that the cell can turn on the genes that it needs when it needs it. Now here's a basic overview of how eukaryotes regulate gene expression. So the first step, and one of the main ways that our bodies can regulate gene expression, is by DNA unpacking. So we're going to learn more about this in a few minutes. But basically, when DNA is all tightly wound up like we see in cell division when we have those X-shaped chromosomes, that DNA is essentially turned off. It's so tight, okay, and so tightly packed that RNA polymerase can't get in there to read the gene. So if our body wants to make proteins or to replicate it, it's got to loosen it up, okay? So that's one of the steps. Once the gene is loose and available for transcription, then transcription can happen. But there are going to be things like those transcription factors we saw right here that are going to help regulate transcription. So these first two steps are actually the main ways that we regulate it. But our body can also control things. So after transcription occurs, you make messenger RNA and it has to be processed. So cutting out introns, okay, and adding the cap and the tail, that can kind of determine not only the code for making the protein, but also the life of the messenger RNA, because there are enzymes out in the cytoplasm that want to eat up that messenger RNA. So the longer the life of the messenger RNA, the more proteins can be made. Okay? Same thing with the proteins that our bodies make. Sometimes proteins don't stay around forever. Sometimes we have enzymes that digest those proteins as well. So in addition to kind of regulating access to the gene, our body can also kind of regulate the messenger RNA and proteins that are made. But for this video, we're going to focus on these two main ways. So let's start off with controlling DNA packing. So this first statistic is pretty crazy. So think about your typical body cell. It's got 46 chromosomes. If you were to take those 46 chromosomes and completely stretch them out, that's about 2 meters or 6 feet of DNA in every cell. Now, depending on your size, you could have up to 50 trillion cells in your body. So that would be 100 trillion meters of DNA. And that's enough DNA to go from the Earth to the sun and back more than 300 times. You could wrap the DNA in your body around the equator 2.5 million times. That's an incredibly large amount of DNA in our bodies. How do we fit it all in? Well, we pack it. So here's the DNA double helix, okay? This is the loose situation, okay? So this is completely loose. This is when enzymes like RNA or DNA polymerase can get access to that DNA. So they can come in, they can do their jobs, okay? So this is the loosest point, and that's when our cells can actually use our DNA. But we aren't using our DNA all the time, so we need to kind of pack it in a little bit more. And what we do is we wrap it around special proteins called histones. So these white, almost bead-like things, those are called histone proteins, okay? And the blue DNA is kind of wrapped around it. Now those histone proteins can then pack further and they can form almost these fibers. And then these fibers can loop up together and condense until you finally get the tightest DNA. And that is when you have mitosis or meiosis. Okay? That's when the DNA is tightly packed so it can be moved and manipulated between cells. Now, we don't necessarily want all of our DNA loose or all of our DNA tight. We kind of want this kind of in between when our cells are doing their normal metabolism. So let's talk a little bit about how our body can actually regulate how tightly packed it is. So there's really two main ways that this can be. There's something called euchromatin and heterochromatin. Okay, so we're going to start over here with euchromatin, which is just a fancy word for loose DNA. Okay, when I say loose, our enzymes can come in, okay, and they can read the DNA, okay? So that means the genes can be accessed. Those genes are turned on, okay? Now, how does our body do that? Well, those histones, those white beads that the DNA wraps around, well, you can add things to those histones to make it so the DNA doesn't want to wrap as tightly. And those things are these A's and these P's. So they're acetyl groups and phosphate groups. Okay, adding acetyl groups or phosphate groups, what they do is they make the DNA they make the histones more negatively charged. And because DNA is negative, because all those negative phosphate groups, the negative histones, okay, don't want to interact with the negative DNA. And so the DNA doesn't wrap as tightly around it. And so you have access, your body has access to the genes. The genes are turned on, okay, and your body could potentially do transcription from it. But sometimes we don't want our genes turned on, in which case we add these things called methyl groups, okay? Methyl groups make it so that the genes cannot be accessed. They're so tight. Those enzymes that need to get in, they get bounced off. They can't get in and read the code. Okay? So we call this really tight DNA heterochromatin. It's essentially turned off. So adding methyl groups to the DNA, that's how you can pack up the DNA, because methyl groups make histones hydrophobic. Hydrophobic means they don't like water, and the nucleus is full of water. So the histones all pack really close together. Okay, they're really tightly packed. So here you can see there's very little space between them because they're trying to get away from the water. And as a result, any DNA that's wrapped around them is going to be really tightly packed as well. So we've essentially shut that off. So this tight DNA, this heterochromatin, that's what's happening in prophase when the chromosome's condensed. Our bodies are adding tons and tons of methyl groups to make it condense down into those X-shaped chromosomes like we see here. So there's a whole special area of genetics that studies these methyl groups and acetyl groups, okay, and how they can be added and taken away from our histones. So we call those tags our epigenome. In Greek, epigenome means above the genome. So remember, genome is just your DNA. That's the double helix. So this is your genome. So adding things to those histones, okay, or sometimes we can add methyl groups directly to that. That is going to be our epigenome. Okay, and so these molecular markers, they're not part of the genes themselves. They just control access to the genes. And the cool thing about these methyl groups and these acetyl groups is they can be added or removed by enzymes throughout your entire life based on your environment. So things like your diet, how much exercise, your stress levels, chemicals you're exposed to, okay, all of those things can add and take away methyl groups and acetyl groups and influence how your genes get expressed. Another cool thing is that we can pass on these markers from cell to cell. So in mitosis, not only are we copying our gene from one cell to the next, we can also copy our epigenome from one cell to the next. And some genes, okay, you can pass on the markers to your offspring for those genes. So you can pass on your epigenome for certain things. Okay, so we can see that our environment can actually influence how our genes work. Okay, so let's say we've unpacked the DNA. We're now at loose DNA. There's a lot of acetyl groups. Okay, everything is available to be accessed. Now we have to actually transcribe those genes. Okay, so to do that, we need the help of transcription factors. So transcription factors are proteins that bind to the promoter, and there's actually two main types. Okay, the first type is an activator. They bind to this region right here that's blue. Okay, that's called the promoter, and it is right before the gene. Okay, and the promoter is where RNA polymerase binds. Okay, so these activators activate RNA polymerase. They help RNA polymerase find the promoter so that transcription can happen. And if you have transcription, you get translation. And if you get translation, you get a protein. So this is truly gene expression. Activator transcription factors help gene expression occur. But sometimes we want to shut off a gene. So there are these transcription factors which act as repressors. And RNA polymerase basically can't get access to the promoter. They bounce right off. So there's no transcription, which means no protein is made. Okay, so this is a gene that's on, whereas this is a gene that's off. Now, this can be way more complicated than just a single transcription factor turning a gene on or off. Okay, The promoter can have multiple transcription factors. It can have multiple activators or multiple repressors. So let's just look at this example here. Okay. So what we can see here is we have two different transcription factors, the green and the blue one, that are activators. When both of them are present, okay, the gene gets turned on and basically the gene strength is really strong. We get lots of proteins, okay? So we're going to see an increase in protein production. But if say only, sorry, I'm running out of room there. If only one of the activators is present, we're going to see that the gene might be on, but it might be weak. And so you might see low protein production. And so it's on, but it's not quite as strong as it was before. And if you got this repressor, this red guy right here, then this is essentially off. Even though the activators are there, it's blocking RNA polymerase, gets repelled. It can't do its job. It's essentially off. So let's put all these ideas together. Okay, there are multiple ways that the cells can turn genes on and off. So let's try to put it all into a common set of rules to help us understand it. So in our bodies, some genes are going to be permanently turned off, meaning in those cells, no proteins are made. Okay, so those are going to be genes that don't relate to a cell's specific function. And in order to turn them off, they're going to be really heavily methylated and tightly wound. So let's take a look at this next picture and let's focus on, say, red blood cells. Red blood cells' job is to transport oxygen. Okay, red blood cells do not make insulin, which regulates our blood sugar. It is permanently turned off. Same thing with myosin, which is a contraction muscle, or a contraction protein found in muscles. It's what helps our body's muscles contract so we can move. Red blood cells don't need that. It's permanently turned off. So you can see those are examples within the red blood cell that might be turned off all the time. Okay. Now, other genes in that red blood cell or in other types of cells in your body, they might be permanently turned on. These are going to be proteins that are always need to be made. They're essential for the cell's specific function or survival. So in this case, we're going to heavily acetylate those histones, and we're going to make sure that they're really loosely wound, okay, so that the cell can access them. And then they're going to have lots of positive transcription factors like activators to make sure that the gene is really on and strong. So let's take a look right here. You can see we call these genes housekeeping genes. So these might be genes that are found, so this blue bar means they're on, and they're on in all cell types. So maybe it's something like cellular respiration genes, right? Every cell in our body has to do cellular respiration to get energy. So that's a really important gene that needs to be turned on to ensure that those cells survive. Okay. So we have some that are permanently turned off. We have some that are permanently turned on. But what about our other option? Well, so some genes have what we call variable expression. They can be turned on sometimes, but they can be turned off other times. So these are going to be genes that relate to the specific function, and then they can be turned on to make proteins or turned off based on the needs of the cell. Okay? So basically, protein production is regulated by temporary binding of transcription factors or temporary winding and loosening. So for example, a gene that's turned on, it might have more acetyl groups, and it might have positive transcription factors at that time. But then the cell wants to turn it off, so it's going to get rid of those acetyl groups. It's going to add more methyl groups, and it might add a negative transcription factor, such as a repressor. So let's take a look here at this pancreatic cell, okay? So let's look at insulin. Insulin is a gene that could vary in expression. So our pancreas's job is to regulate blood sugar, okay? Insulin's job is to decrease sugar in our blood. So our body really only needs our pancreas to make it when we have a lot of sugar, Okay, so when we have high amount of sugar, our pancreas can sense this and it can turn on the gene to make insulin. So high sugar would mean it's on. And that would tell your insulin would be produced, and insulin would then tell your cells to go ahead and use that sugar, and so that we can maintain homeostasis and keep the right amount of blood sugar. But when we have low sugar, we don't want our sugar levels to drop even more, so our body turns it off. Okay? So this is an example of variable expression. Okay? Based on the needs of the cell, it might be on or it might be off. And so our bodies are really going to be kind of maintaining this winding and this transcription factor in the right balance to ensure that we can do the right things at the right time. Okay, because our ultimate goal is for gene expression to maintain homeostasis and to keep our bodies alive.